Welcome to Scrubcast, where we take a closer look at the research happening at Stanford University's Department of Surgery. I'm your host, Rachel Baker. Today, we're speaking with Dr. Nick Leeper. Welcome to the show.
Thanks, Rachel.
Thank you for joining us. Dr. Leeper, you are Chief of Vascular Medicine here at Stanford and a professor in our Division of Vascular Surgery. But you are not a surgeon, so I'm just going to ask the awkward question and get it out of the way. Why are you in the surgery department?
That's a great question. So I'm actually a board-certified cardiologist, but I actually am in love with blood vessels outside of the heart. So I do a subspecialty of cardiology called vascular medicine. And you can kind of think of it as a hybrid between cardiology and vascular surgery. And, when I came through my training years and years ago, I won't say how many years ago, my research was much more naturally aligned with what was going on in the Division of Vascular Surgery. So I joined the Department of Surgery to help, build this bridge and this interface between internal medicine and surgery. And we've had a lot of success trying to improve the care of patients with vascular disease and then, do research related to their underlying conditions.
Fantastic. Well, we appreciate that you're in the Department of Surgery. You did your residency in internal medicine. You completed 2 fellowships, one in cardiovascular and one in vascular medicine. But what drew you to vascular in the first place? What was it about blood?
Well, it's more, I think, about public health and, what are the world's leading killers and where can we have the largest impact. Cardiovascular disease has long been the leading killer in the Western world. It's overtaken infectious diseases as the leading killer worldwide. And most cardiovascular disease, like heart attack and stroke, is driven by atherosclerosis, which is the buildup of plaque in the arteries. And so when I was trying to decide what I wanted to do when I grew up, I thought I wanted to work on something that was important, that could help a lot of people if we had success. And so that's why I chose to work on cardiovascular disease at a large scale and why I studied this particular process in particular.
Awesome. Well, so one of the things your lab specializes in is genome-wide association studies, or do we pronounce this GWAS?
Yeah.
Oh, excellent. Okay, so what does that mean?
It's a fancy term that really just applies to a case control study, but one that's applied across the entire genome. And so, what do I mean by that? Well, if you think about our DNA, we have 3 billion base pairs in every cell in every part of our body. Most of that DNA is pretty similar from person to person, even species to species. The A's, T's, and C's and G's are pretty common across people. So it's actually a very small percentage of our DNA that makes us different. Tall versus short, black eyes versus brown eyes, or diseased versus healthy. And so the power of these genome-wide approaches is we can get large populations of patients, both healthy controls and people with the disease of interest. And now the sequencing technology has gotten so affordable that we can very rapidly sequence all 3 billion base pairs.
Okay, I wanted to ask you about this.
Yeah, sure.
The original Human Genome Project, it took 13 years to sequence a genome. How long does it take you?
Oh, I mean, it's almost free. I mean, it was we send them out and they come back in a matter of days. We can do dozens of samples at a time. You're right. I mean, the Human Genome Project took, like a decade, billions of dollars. Now it's, less than $100 and comes right back.
Wow.
Yeah. And so, the real power here is because people have made investments in doing this across large populations of patients. 500,000 people in the UK Biobank, 1 million people in the Million Veterans Project through the VA. And so when you have these really large samples, you can start to make collections of people with or without a disease. And then you can find these genetic variants, which are more commonly found in people who have had a heart attack versus not. And that gives you the clue about what gene or pathway to go after. And so this is really powerful. It's unbiased. It means that we're not chasing a hypothesis. We're saying, you know what, let's look across the entire genome and then let biology tell us where to look next.
Okay, that was my next question. Is you say on your website that you're hypothesis free, so you're just throwing spaghetti at the wall and kind of seeing what sticks and then being like, "Oh, okay, I see an association here. Like, let's explore that."
Yeah, that's exactly what we do. We are not really all that sophisticated, to be honest. And in fact, it's kind of intentional. We do not want to work on the things that we already know about. We already know about LDL cholesterol. We already know about high blood pressure. We already know about diabetes. And I'm not saying that we don't have a lot of work left to do to further improve cholesterol management and diabetes management. But we know that even if you could fix all of the traditional risk factors, that only about half of our risk for disease would be addressed. So we are very much interested in the unknown. What is that other half that's either inherited or through environmental exposures, our behaviors? What are these things doing that we can't yet address with current medicines? And so that's why we say we want to intentionally look away from those and let the genome teach us what else we've been missing so far. And it's been really shocking because it's things that we wouldn't have thought had any relevance to heart disease or stroke. It's really things that we're learning from the field of oncology or immunology, and it's really having to change the way we think about disease.
Super cool. So, I want to dive into this paper that you authored in Nature Communications this past September. I'll put a link for our listeners in the description. Okay, you've defined one of the terms that I think is important, atherosclerosis,
Yeah,
buildup of plaques. We've talked about this on the podcast before. But then there's another term that you use in the paper, and it's apherocytosis. And that's the cleanup crew?
that's right.
Okay. Atherosclerosis happens when the apherocytosis isn't working. There's an imbalance.
Yeah, yeah. I mean, it's a pretty complicated process, but atherosclerosis is the process that builds up in our arteries as we age. This is the plaque. It's composed of cholesterol and inflammatory cells and all kinds of other gobbledygook that gets clogged up there over the years. By the way, this starts in our teenage years and takes decades to develop, like a slow growing tumor. And there are a lot of reasons why atherosclerosis can progress. It's not one cause, but we believe that part of the problem is that our immune system can't do its normal job and can't go after and get rid of this. We know that many cells in our body have the ability to identify this inflammatory stuff, these dying cells, these necrotic cells, and they should actually eat them and get rid of them, almost like Drano within the plaque.
Cool.
But again, because of genetics, we know that certain people are born with defects in these mechanisms, and then the immune system will kind of ignore this tissue and allow it to continue to accumulate over time. So we're very focused on trying to find ways to reactivate that and to get our immune system to basically take out the trash and remove all that built up plaque that we know leads to heart attacks and strokes.
So you're saying no matter how many cheeseburgers I don't eat and how many miles on the elliptical I run, if my genetics are bad, I'm probably still going to end up with some buildup in my arteries.
Yeah, I mean, if you picked your parents poorly and didn't plan ahead, that's absolutely the case. I mean, for sure, we always start with education about lifestyle, diet, exercise in particular. But you can only move the needle so far with those. And for a lot of patients, we need to be on aggressive cholesterol lowering medicines. And now we think we need to find ways to tackle these other aspects to get at what we call the residual risk that's not currently addressed.
Okay, so you were talking about our immune system is supposed to go and eat
Mhm.
the plaque and the necrotic cells and things. And so you identified a molecule that's called CD47, and you call it the "don't eat me" molecule. Is it like my plaque has just like figured out a way to be like, "Oh, I'm staying here. Don't evict me. I'm a squatter."
Yeah, we think that's exactly what's happening. And it's really fascinating because this is the same type of process that cancers use as a way to escape the immune system. It's also a thing that invading bacteria use to try to escape, removal by our inflammatory cells.
Hmm.
So we think that this gets turned on in a pathological way. And I kind of think of it as like a cloaking device. So there's the plaque, the same way
Hmm.
that there's an early cancer. And normally, our immune system is smart enough to say, this is non self. We should get rid of it. This is dangerous. This is going to cause trouble. Let's get rid of it while we can. But it turns on these signals that basically make it invisible to our immune system. And they're actually called these funny terms like don't eat me signals. And so they basically help these cells escape the immune surveillance that would normally be happening. And then they're allowed to continue to grow over time.
A side question: are you a Star Trek fan?
Yeah, Star Wars more than Star Trek, but anything
Oh,
with aliens or explosions generally
okay. Well, you
is fine
mentioned a
with me.
cloaking device, so I'm like,
Mhm.
are we calling the plaques the Romulans here? So,
Yeah.
anyway, so in an earlier study, you cited it that you said systemic blockage of CD47 resulted in anemia, and that's when you don't have enough iron in your blood. So obviously, we can't just turn off CD47 systemically. Is that the problem here? And so you're trying to overcome a that challenge.
Yeah, I mean, so a lot of what we've done since we figured out that this is the problem at baseline is we say, okay, well, how do we reverse this? How do we turn the immune system back on and make it start to engulf and get rid of all these cells that we don't want? And we can do that. It's not complicated. You just block the don't eat me signal, you take away the brake, and then all of a sudden the gas can go faster. But the problem is that the therapies we have in the first generation are a little bit too good.
Oh.
They get, yeah, I mean, it's true. I mean, we can get rid of the cells we don't want in our plaque, but we also get some removal of healthy cells.
Oh.
And as you mentioned, the red blood cells are particularly susceptible for reasons we don't fully
Huh.
understand, but they will get gobbled up. And so while your plaque is melting away, or your cancer, if you're an oncologist, unfortunately, the red cells get eaten up too, and then you get anemia, which is a side effect that we really, can't accept
Mhm.
for, thinking about applying this to lots of people with heart disease out there.
Got it. Because I mean, I assume that it's anemia that's so bad that eating a little bit of red meat and some spinach is not going to help me.
Well, in some cases, we worry that we'd have to transfuse patients, and so
Oh,
that's,
oh,
you know,
that's bad.
yeah, it's probably worse than what we would want. Now, we have a lot of efforts trying to understand this biology better to make a better mousetrap and find new ways to get the clearance where we want it, but to leave the healthy tissue alone. One of the things we've done is we partnered with our colleagues at the bioengineering school here, and we've been making what we call our Trojan horse nanoparticles, where
Oh,
we can have basically a nanotherapy that will only go to the diseased blood vessel. And so now what we can do is we can deliver our very powerful therapy where we want it, but we're not having it sent elsewhere. And so we've shown that this will lead to the benefit we want, but we don't get those side effects. We're not seeing the anemia, we're not seeing the clearance of other healthy cells.
interesting.
And so we think that this could be a way to have our cake and eat it too, and really have the benefit without the downside.
So, are the nanoparticles like the ones that we saw in Star Trek? Like, yeah, I get an injection, and all of a sudden there are all these little tiny bots flying through my bloodstream and being like, "We're going to the heart." And like, how do they know that? How do you program them to do that?
That's how I think about them. And we actually had an artist design a beautiful drawing that's exactly along those lines. We're still trying to understand why they are so specific for the inflamed cells. We don't really know why they're gobbled up there, but we love the fact that that's where they accumulate. And so we can give very high concentrations of our therapies, which we basically stick on the side of these nanoparticles without having them go elsewhere. And so it's exactly right. We give these intravenously. We've shown that they have potential in mouse models. We've shown they have potential in large animal models. And we're hoping that, one day they'll be in humans as well.
And so you partnered with someone from BioE to do this. Do you just like wander over there and be like, "Hi, I've got a problem." How does one do this? I've heard about how Stanford is like this slippery slope of collaboration. Like everyone just wants to collaborate with everybody else, but I don't know the first. Maybe it's because I'm an introvert, but I would not know where to start.
Well, I mean, that's, one of the real things that's magical about Stanford is that there are these traditional silos that we're used to really don't seem to apply. If you have a clever idea, you can go knock on the door of any of the Nobelists here or anybody that's a department chair from a different division or section. And my experience is they've always been open to clever ideas. and the power of bringing a different viewpoint and bringing a different background and integrating our skills to tackle problems through a new rubric. This is one of the things that makes Stanford so special. And I've never been told no so far, so it's a great thing about working here.
Amazing. Well, I absolutely want to keep talking about this, but that sound means that it is time for our lightning round. On each episode of Scrubcast, we ask each of our guests the same 3 questions, and the first one is, who is a surgeon you admire and why? I don't know. Do you have surgeons that you admire?
Yeah, I mean, I think the person that made the biggest impression on me was Norm Shumway. He was still practicing when I was a fellow here,
Really?
and I've never seen somebody that made such pioneering advances that was so humble and so willing to invest in the next generation of trainees. He really wanted to pass on his legacy. And I just admire his perseverance, inventing, the concept of the heart transplant and implementing it in the United States for the first time, thinking through all the technical issues, the immunosuppression, figuring all that out, and really, really never giving up was very inspiring to me.
Absolutely. 100% agree. Norm Shumway. The second question we ask is, what is the best advice you have received in 10 words or fewer?
10 words. Well, this probably comes from our last chief at vascular surgery, Ron Dalman, who, pretty early taught me that, how does it go? Hard work beats talent when talent doesn't work hard. Um, and I like that because science is hard. It's hard to begin with just in general, but if you're tackling a big problem and you really want to challenge paradigms, you're going to get a lot of headwind. And this concept of settled science is one that's really hard to surmount. And I think that you have to be willing to not only do the hard work in the lab, but accept rejection, not feel bad for yourself. Understand that people are going to initially resist the ideas in your grants and your papers, but sticking with your beliefs and persevering, this is where you see the enormous success from people like Shumway and the impact it can have. So I think that, just perseverance and having a never give up attitude was something I was glad that Ron taught me in my early days.
Love it. I'm going to add that one to my wall. Well, in our last episode, we started asking our guests about their OR music preferences. Is there such a thing as lab music? Is that a thing?
Oh, sure. Yeah. We, in my lab, we like both kinds of music.
There are 2 different kinds? Only 2?
Yeah. Only 2. Yeah.
What are the 2 different kinds of music?
Oh, you haven't seen Blues Brothers, I guess.
No, I'm sorry. I'm okay. I'm showing my age.
Well, the classic is we like both kinds of music, country and Western.
So a lot of Blake Shelton going on in your lab?
Sure. We'll do it all. Yep. No, it's our lab is really wonderful because we have people from all over the world, so it really is a melting pot, and we get to enjoy all the different cultures in our lab, and so we are always trying out new stuff.
Fabulous. Well, it has been a pleasure chatting to you. Before we go, one last question. What is next for Dr. Leeper?
Oh gosh, we are now really moving beyond atherosclerosis alone and starting to look at the interface with cancer and cardiovascular disease. We've known for a long time there are many shared risk factors, but we think that there may be more than that, maybe direct causal relationships. And so we think that if we can study this interface, we may be able to address both the number 1 and 2 leading killers in the world. And we're really excited to embark in a bunch of new efforts that we're starting in the lab at the moment.
Fantastic. Well, I look forward to seeing what you come up with. Thank you so much for joining us on Scrubcast.
Thanks, Rachel.
And thank you to our listeners for tuning in to this episode of Scrubcast. Until next time, stay sharp. And that brings us to the end of another episode. If you like Scrubcast, we hope you'll tell your friends and subscribe wherever you get your podcasts. Scrubcast is a production of Stanford University's Department of Surgery. Today's episode was produced by Rachel Baker. The music is by Midnight Rounds, and our chair is Dr. Mary Hahn.